All right, BIM 100 students, let's talk finals. Specifically, let's talk about Lecture 4. We all know this is the tricky one, right? It's less about just memorizing facts and way more about thinking through experiments and concepts. It can feel a little daunting, but don't worry. Today, we are going to crack it wide open. So here's our game plan. First, we're going to tackle the mindset for Lecture 4. Then, we'll walk through how to actually read an experiment, how to predict what happens when things go wrong, and of course, how to nail down those super important replication details. And we'll wrap it all up with a final study tip that's going to tie everything together. Let's do this. Okay, so what's the real secret for this part of the final? It's not just about knowing what happens. It's about understanding how we know it and why it matters. You have to start thinking like a real molecular biologist, and that's the exact skill we are going to focus on building right now. I mean, try to see it from Professor Pescanelli's point of view. She wants to know if you can connect the dots, Can you look at a few simple bands on a gel, on a southern blot, and from that, figure out something as big and complex as the chromatin state of an entire gene? That's the leap. And that's exactly what we're going to practice, going from that raw data to the big biological story. All right, let's dive into our first big skill, reading an experiment. And we're going to use the DNase protection assay as our main example. It's a total classic from the lecture and, a very common type of exam question. Okay, just picture it. You flip the page on your final and bam, there it is. A gel, a bunch of lanes, a bunch of bands, and it is so easy to panic just a little bit. But stop. Take a breath. Before you even start analyzing, what is the single most important thing you should look for? What's the one comparison that unlocks the entire puzzle? So let's just quickly run through the logic here. First, you gotta isolate the nuclei. That keeps the chromatin all bundled up and intact. Then you hit it with DNSi. That's the real test, right? To see what DNA is accessible and what's hidden. After that, you purify whatever DNA survived. You chop it up with some restriction enzymes, and finally, you use a southern blot with a very specific probe to hunt for your gene. It's really like a little detective story, just one step at a time. Okay, now let's see what this looks like on an actual exam question. See that normal lane? Look at the control, the one without DNase. That's our baseline, a nice, strong band. Now, look what happens when we add the DNase. That band is still there. What does that tell us? It tells us the DNA was protected. It was wrapped up super tight in heterochromatin. But now, check out tumor 3. When you add DNase, poof, that band almost completely disappears. That means the DNA was wide open, totally exposed, and the DNase just chewed it up. That's our euchromatin. See? The entire story is right there in that plus versus minus comparison. Now, let's make sure you don't fall for some of the really common traps here. Pro tip number 1, the size of that band you see is determined by where the restriction enzymes cut, not by the size of the gene itself. That is a classic trick question. Pro tip 2 and 3, just lock these in your brain. Condensed, closed-off heterochromatin goes with histone methylation. Open, accessible euchromatin goes with histone acetylation. If you can get those two things straight, you're grabbing some easy points for sure. Okay, so we've just figured out how to interpret what an experiment is telling us. The next level up is being able to predict what the data would look like if something broke. This is another one of those favorite exam question styles, the what-if scenario. And for this, we're going to use human artificial chromosomes as our perfect model. First, just a super quick refresher. A human artificial chromosome, or a HAC, is basically a man-made chromosome built with just the 3 absolute essentials needed to work. And you can bet the exam loves to ask what happens if you leave one of them out. This table right here lays out those what-if scenarios perfectly. So, what if you take away the origin of replication? Well, the chromosome never gets copied, so after the cell divides, only one of the 2 daughter cells will get it. That's where the 50% comes from. What if you lose the centromere? Well, it replicates just fine, but the cell has no way to pull the copies apart properly. It becomes a total crapshoot. Some cells might get 2, some might get none. And if you get rid of the telomeres, you've lost the protective end caps, and the chromosome literally starts to fall apart and can even fuse with others. Each failure tells you exactly what that part was supposed to do. All right, time to switch gears to our third key skill, absolutely nailing the details of the molecular machine. When you get questions about DNA replication, it really is all about knowing which enzyme does what job. So here is your rapid-fire guide to the major players. You've got helicase, that's the unzipper,
You've got primase, that's the RNA starter, laying down those crucial little primers. And then you have the main builders, DNA polymerase epsilon, who has the easy job of building the leading strand, and DNA polymerase delta, who has the much trickier job of building the lagging strand.
This comparison table, this is everything. The leading strand is a simple one, right? One primer, continuous building by Paul epsilon, and it just follows the replication fork. The lagging strand is the complete opposite. It needs a ton of primers, it's built in those discontinuous chunks called Okazaki fragments, it's made by Paul delta, and it moves away from the fork. This table is your absolute go-to for any question that asks you to compare these two. So, okay, we know the lagging strand is complicated, it's messy, but let's connect that fact to a much, much bigger idea. How does this weird backwards process actually create a huge fundamental problem for the cell, the thing we call the end replication problem? And the answer is actually pretty simple, but it is so critical. When that very last RNA primer on the very end of the lagging strand gets removed, there's nothing for DNA polymerase to build from. There's no 3' hydroxyl group to grab onto, so that little gap just can't be filled in. And this means that with every single round of cell division, the chromosome gets a little bit shorter. And that is exactly why telomeres are so absolutely crucial. They're the buffer zone that protects our actual genes from being lost. Okay, let's bring this all home. We've gone over how to interpret data, how to predict outcomes, and how to master the details. Now it's time for that one final tip that really connects everything we've talked about. This right here. This is the whole key. You have to move beyond just being a flashcard robot. For every single concept you study, you need to actively ask yourself 2 questions. How does this experiment actually prove this mechanism? And what would happen to the entire system if this one little piece just failed? It's that deeper, more analytical way of thinking that is going to make all the difference on the exam. So, I'm going to leave you with one final challenge. Take this exact same way of thinking and try to apply it to another topic. Maybe something like RNA splicing from Lecture 10. If splicing goes wrong, what would that look like on a northern blot? If you can start reasoning your way through problems like that, you're not just ready for Lecture 4. You are ready for this whole final. You've totally got this. Good luck.